Now that we have a basic understanding of what molecules are, we can delve into organic molecules, which are the molecules of life. So, organic molecules are mainly carbon based molecules, and organic molecules are essentially the molecules of life, which includes carbohydrates, proteins, lipids, and nucleic acid. They they are mainly associated with living organisms. So their main source, or they're found in the living systems. These biomolecules help us with our understanding of how the life kind of originated on Earth. So primitive Earth, about 4 billion years ago, had very harsh environmental conditions. The reducing environment was rich in gases like methane, ammonia, hydrogen, and water vapors. And there were intense energy sources such as lightning, volcanic activities, and UV rays. So all these energies brought about the formation of molecules. So here in the reaction, you can see that how small molecules have now been able to combine and make big molecules, such as the on the right you see the formation of amino acids from methane, ammonia, hydrogen gas, and water. Let's talk about Miller and Urey experiment of 1953. The objective of this experiment was to test whether organic molecules could form under Earth's early environmental conditions. So what they did is they prepared a closed system with the gases such as methane, ammonia, water vapor, and hydrogen gases in a closed container, and then they sparked electrical spark on them as a source of energy, and they also heated water to create water vapors and then condensed them, simulating a kind of a water cycle. The result of this experiment was that within a week, organic molecules were formed, and they could collect some amino acids, and amino acids, are the building blocks of proteins. And they demonstrated that simple organic molecules could combine when provided enough energy, and they can start synthesizing larger organic biomolecules. Miller and Urey's experiment supported the hypothesis that life's building blocks could form naturally from the inorganic material. So inorganic molecules like ammonia or methane or water could combine to form amino acids, and amino acids are biomolecules. For this to happen, there has to be the correct kind of environmental conditions, which leads to the theory of chemical evolution. Basically, chemical evolution means that the life actually started from chemicals. So there were smaller molecules in the beginning, and these smaller molecules combined over time to form larger molecules and even more complex, huge biomolecules. And these biomolecules then aggregated in these droplets or co-servates that started the formation of first primitive cell. So the modern view says that organic molecules might have formed in deep sea, which are hydrothermal vents, and through some extraterrestrial sources like meteors or some kind of radiations, these organic molecules could combine and form complex biomolecules, and these combinations of biomolecules then led to the formation of primitive cell. Then let's talk about these important organic or biomolecules, which eventually will lead us to the story of the structure of cell. So organic molecules are broadly classified into 4 major categories: carbohydrates, lipids, proteins, and nucleic acid. Carbohydrates are organic molecules which contain carbon, hydrogen, and oxygen, generally in the ratio of 1:2:1. Carbohydrates are the primary source of energy for living organisms. So carbohydrates are the first class of nutrients that we digest or burn to obtain energy when we need energy to do work. So they are the main providers of energy. They provide structural support, and they are also used as a storage molecule for emergency use of fuel. The simplest form of carbohydrates are monosaccharides. So monosaccharides is one unit of sugar, one molecule of sugar. And as we talked previously, carbon, hydrogen, and oxygen are in the ratio of 1:2:1. So here you can see that in glucose you have 6 carbons, 12 hydrogens, and 6 oxygen atoms. This is the structure of fructose, this is the structure of galactose, and all these are one sugar molecule or a single sugar unit. They all are monosaccharides. So glucose is extensively found in our blood, and fructose is found in fruits, and galactose is a component of lactose, and lactose is the milk sugar. When two units of single sugar molecule combine, they form di, means two. Di means two, so they form disaccharides. For example, glucose and fructose combine to form sucrose, and sucrose is the table sugar that you use in your kitchen. Glucose and galactose combine to form lactose, the sugar that's found in milk. And two molecules of glucose form maltose, and maltose is present in grains from which syrups are formed. Now poly means many. So mono means one, di means two, and poly means many. Monosaccharide means one unit of sugar. Disaccharide would mean two units of sugar combined together, and polysaccharide means many units of single sugar combined to form a complex molecule of sugar. So polysaccharides are of different types. First type is starch. Starch is the storage sugar of plants, and it is found in high concentration in potatoes, grains, and corn. The molecule of starch is broken down into glucose by the amylase present in our alimentary canal and the salivary amylase present in our saliva. Another important example of polysaccharide is glycogen. Glycogen is this highly branched structure formed by coming together of glucose molecules. Glycogen is the stored form of sugar or stored form of glucose in human beings. It is stored in liver and muscle cells in human body. It is quickly mobilized as glucose when our body needs energy, and it acts as a short-term storage reserve, but eventually it's used to balance out when our body or our blood sugar is dropping. Chitin is another important polysaccharide. Chitin is what forms the exoskeleton of the insects like crabs, shrimps, lobsters, and they also make the wings of insects. So this material is tough, and it has strength, it has flexibility, and it is also resistant to degradation. That's why it can make the external structures of insects. Another very important polysaccharide is cellulose. Cellulose is formed by the linear chains of glucose, and cellulose is what makes the cell wall of plant cell. So cellulose provides rigidity and strength to plant. It's insoluble in water, it's not digested by human beings, and in herbivore animals they have bacteria in their gut that helps them digest cellulose. So now you have seen that the single units of organic molecules can combine to form long complex structures. So this joining of monomeric units or single units to form complex multimeric structure or polymeric structure, this process is called polymerization. And in this example, you can see that one unit of OH and one H from here is removed. So this is H2O, water is removed from this reaction. This is called dehydration because of removal of water, and these two molecules can then form a chemical bond. But when you add water to it, so this water is added back, then this bond is broken and you have reverse reaction, which is called hydrolysis. So formation of chemical bond in monomers to form polymers is called polymerization. The next class of biomolecules is lipids. Lipids are also hydrocarbons. They contain carbon, hydrogen, and oxygen, and they are hydrophobic in nature. They are insoluble in water, but they are soluble in non-polar solvents, such as oils. The one most common or basic kind of lipid is fats, oils, which are collectively called triglycerides. Why are they called triglycerides? Because they have one molecule of glucose and three molecules of fatty acids. So this is a glycerol, and this long chain of carbon is a fatty acid. So three molecules of this and one molecule of this combine to form a triglyceride. So these triglycerides are your oils and fats and butters that you use in your kitchen. The fats can be saturated or unsaturated depending on whether they have a double bond in their structure or not. If they have no double bond, that means this structure is completely saturated with hydrogens. Therefore, they don't need any more hydrogen. They have no double bonds. One molecule of this fat can stack over the other molecule and another molecule. So this stacking together makes them solid at room temperature. Whereas the unsaturated fatty acids have a double bond, so they are not completely saturated with hydrogen. You can still add one hydrogen here and one hydrogen here, and they, because of the presence of this double bond, they have this kinky structure, so they cannot be stacked one on top of other. That's why they are liquid at room temperature. So fats are used as the long-term energy storage in our body. They provide insulation to our body, they provide protection to our body. Biologically, saturated fat are unhealthier to eat because they get accumulated in our arteries and can lead to the blockage of arteries, and unsaturated fats are considered more healthier version of fats for our diet. Another form of lipids called the phospholipids are also very important. As I told you, one glycerol and three fatty acids make a molecule of triglyceride or lipid. If you remove one of these fatty acid chains and add a phosphate instead of that, it becomes a phospholipid. These phospholipids have this structure where you have one glycerol, two fatty acid chains, and a phosphate. This structure or this molecule is very important because it is the major component of plasma membrane. If this molecule had not existed in nature, we would not be able to create a boundary material or the wall material for a cell that encloses all the essential material inside for the formation of first cell. So interesting thing about its structure is that the head region is hydrophilic in nature and the tail region is hydrophobic in nature. So it makes a bilayer where these stacks are standing kind of next to each other with hydrophobic moieties or hydrophobic tails inward and hydrophilic heads outward. So this side and this side of this molecule can still interact with water or aqueous material around them, whereas the lipid or the hydrophobic part is embedded inside. Steroids are another important form of hydrocarbon which contain these four ringed structures, and these steroids are very important in our body for different functions. Some of example of important steroids are cholesterol, which is a structural component of our cell membrane. Then we have testosterone, which is a hormone, and estrogen, which is a important hormone for females, and they provide sexual characters to male and female. And then we have cortisol, which is another hormone, and it is a steroid hormone, and it's involved in our stress response and metabolism. Steroids play a crucial role in our body. They regulate physiological processes like metabolism, immune function, and reproduction. Especially when it comes to cholesterol, it really helps with maintaining the fluidity of plasma membrane, making sure that our cell membrane stays flexible, is functional, and it is tolerant to variations or changes in temperature. The next class of biomolecules we are going to discuss are proteins. Till now, we have learned that carbohydrates and lipids essentially contain carbon, hydrogen, and oxygen. But in proteins, another very important atom is required, which is nitrogen. And nitrogen is what makes the amine group of amino acids. So amino acids are the building block of proteins. Amino acids link together through a peptide bond to form a long chain called a polypeptide chain. This polypeptide chain is called the primary structure of a protein. But if it becomes a helix or becomes like a folded sheet kind of structure, then that gives a secondary structure to the proteins. The tertiary structure is when alpha helix or beta pleated sheets are further coiled over themselves and make a 3D structure. When this 3D structure protein gets associated with other polypeptide chains, that makes the tertiary structure of a protein. But a protein can have a tertiary structure or is just enough to have one properly folded chain, depending on what kind of function protein is playing in our body. So different kinds of proteins can have different kinds of structures and may or may not be associated with other polypeptide chains. So proteins are mainly classified as structural or functional. There can be structural proteins or functional proteins. Proteins are very, very important molecules in your body. They make structure in your body. They perform various kind of functions. So although it's a nutrient in your food, but once it has been integrated into the different parts of your body, it is the last source of nutrient that you would want to digest. So you would want to digest your carbohydrates and fats first, and then finally the body goes into digesting the proteins of the body. But if you are digesting the proteins of your body, it can lead to severe malfunctions of different organs. So some example of some structural proteins are keratin. So keratin protein is present in your hair. If you have good amount of keratin protein, it ensures your hair are healthy. Collagen is under your skin. As you age, collagen fiber or collagen protein degrades, and that leads to wrinkling of skin. Actin and myosin are present in the muscle fibers, and interaction of these actin and myosin proteins leads to contraction and relaxation of your muscles. Tubulin form the cytoskeleton, means they form the skeleton inside your cells that provides shape to the cell and also helps with transport of different material from one part of the cell to the other. Functionally, proteins can be enzymes which catalyze several reactions. For example, here we are looking at an enzyme that is catalyzing the polymerization of glucose into maltose. Proteins can be hormones. For example, insulin is a protein, and insulin helps regulate the blood glucose in our body. Protein can also play a role in transport. For example, hemoglobin is a protein that transports oxygen, and it's present inside the red blood cells. Functionally, protein also play a crucial role in our defense mechanism, in our immune system, because the antibodies are basically proteins. They are immunoglobulin proteins which attack the foreign pathogens and protects your body from infection. So how are proteins formed? Proteins are formed from amino acids, which are the building blocks. Amino acids are again carbon, hydrogen containing molecule that has an amine group and a carboxylic group on the other side, and it makes a bond between amine group of one molecule and carboxylic group of another molecule, and they make a bond, and this bond is called the peptide bond, and this long chain of amino acids is called a polypeptide chain. Next class of biomolecules that we are going to discuss is nucleic acid. So nucleic acid are the macromolecules which are related to the genetic regulation. They control your genes. Actually, they make your genes, and genes are what define you, how tall you are, what is your complexion, what is your hair color, what is your eye color. Everything is defined by your genes, and genes are nothing but the strands of DNA. So nucleic acid are of two types, DNA and RNA. DNA is the molecule that contains the genetic information. It's present essentially inside the nucleus of your cells. Structurally, both DNA and RNA are made up of nucleotides, and nucleotides are made up of sugar plus phosphate plus a nitrogen base. And we'll talk about that more in a minute. So DNA is like an instruction manual for your cell. It encodes all the genetic information that is needed to build and maintain an organism. It's also passed down from parents to offsprings, and that's how you maintain the continuity of your genes. RNA is involved in transcribing the message of DNA and converting them into proteins, and it's the proteins that ultimately lead to all the characters in your body. If you have melanin in your skin, melanin is a protein, your skin is dark, and if you have less melanin in your body, your skin is light. So in that term, your skin complexion, your hair texture, your height, your everything is governed by proteins, and protein, the information about protein is stored in the DNA. So nucleotides are the building blocks of nucleic acids. So what is a nucleotide? This one molecule is a nucleotide. It has a 5-member sugar and a phosphate molecule and a nitrogen base. Now, what is a nitrogen base? The nitrogen base can be this molecule, or this, or this, this, or this. So what you're seeing here is the nitrogen bases can be double ring structure or single ring structure, and they are adenine, guanine, thymine, and cytosine in DNA. And in RNA, you have adenine, guanine, cytosine, and uracil. Now let's look at the structure of DNA. Here you have a 5-carbon sugar. This is sugar, this is sugar, this is sugar, and this is sugar. And you have phosphates in between them. So it's a sugar phosphate backbone. On the other side also you have a sugar phosphate backbone, but you'll see that O's are on the lower side here and O's are on the upper side over there. So this chain is running this direction and this chain is running in this direction. So we say this chain is running in 5' to 3' side, and this is running from 5' to 3' side on the other side. And these strands are anti-parallel. Here you have nitrogen bases, and these nitrogen bases are making pairs, or they are connected through the hydrogen bonds. So now how are these pairs formed? The pairing is always between A and T and C and G. Here also it's A and T and C and G pair. So a simplified structure is you have a sugar phosphate backbone and there is another sugar phosphate strand which is running anti-parallel. So it's a double-stranded anti-parallel structure. These two strands are held together through hydrogen bonds between nitrogen bases, and these bonds are formed in a way that A pairs with T and G pairs with C. Once this ladder has been created, it gets coiled, and finally you get this coiled DNA structure. RNA is a nucleic acid which is mainly involved in formation of proteins. So I told you functionally proteins are really important. Proteins are what defines all the characters and perform several important functions in your body. But for the proteins to form, there is a code book that has to be read. That code book is called DNA. And who reads that code book? RNA. So RNA is the code book reader or the decoder of DNA for helping the formation of proteins. So the ribonucleic acid or RNA plays an important role in communication and expressing the code of DNA and manifesting the information that is actually written in the form of a code in DNA. There are different kinds of RNA in a cell, and they are involved in different steps in the process of protein synthesis. If you talk about structure, then DNA is a double-stranded structure, but RNA is a single-stranded structure that has a sugar phosphate backbone. But the difference is in DNA you have deoxyribose sugar, and in RNA you have ribose sugar. Means here you have one less oxygen, and here you have oxygen. Also, it contains nitrogen bases, but instead of thymine, as in DNA, it has uracil. So this kind of become identification feature for whether you are looking at a DNA strand or an RNA strand. If the strand contains uracil, you know that you're looking at an RNA strand. And if there is thymine in the strand, then you know that you're looking at a DNA strand. There are several different kinds of RNA present in the cell, but majorly three forms: the messenger RNA, transfer RNA, and ribosomal RNA. The messenger RNA carries the genetic information from DNA to the ribosome for protein synthesis. The transfer RNA plays a role in gathering amino acids from which the long chain of proteins are formed, and so you can imagine it plays a very critical role in the formation of proteins. And the ribosomal RNA actually are the structural unit of a ribosome. So ribosomal RNA and riboproteins make a complex that forms the smaller and larger subunit of ribosomes, about which we'll talk more in coming lectures. So here you know that ribosomes are involved in the formation of a very important cell organelle which is called a ribosome. Now let's quickly recap the differences between DNA and RNA. Both DNA and RNA are made up of nucleotides. So nucleotide is a sugar, phosphate, and nitrogen base containing molecule. That means sugar phosphate backbone is present in both RNA as well as DNA. But there are two strands of this sugar phosphate backbone in DNA and one in RNA. Then these lines that you're seeing are representing the nitrogen bases. So the nitrogen base present in RNA are adenine, guanine, cytosine, and uracil. And the nitrogen bases present in DNA are adenine, guanine, cytosine, and thymine. Further, the backbone forming sugars are ribose sugar in RNA and deoxyribose sugar in DNA. That is why their names are slightly different.